and today I'm going to be talking about the scanning electron microscope and how it can be used in forensic science as well as other sciences. So some background on the scanning electron microscope and energy dispersive X-ray analysis combination is that this is a combination of a microscope and an X-ray technique that can tell the scientists about the morphology, which means like the physical characteristics, how the sample looks, and the elemental composition of a sample. If you look at the image here on the left, the very left side of the image shows the morphology of the sample, and the right side shows the results of the EDX analysis, which results in a spectra. And each one of those peaks correlates to a specific element, and these spectra can be used to determine the elemental composition of a sample, as well as elemental mapping for the location of those elements within the sample. And as you can see, the left side, the images on the right, are high resolution images of the sample at super high magnifications. And they can tell a lot about the crystalline nature and the cellular structure and characteristics of that sample. So the combo SEM EDX can be used for both microscopic and elemental analysis, like I was just talking about on the last slide, where the microscopic analysis provides the picture of the sample, and the elemental analysis is done by the EDX attachment, where the composition of the sample and the elemental mapping is completed. So how does the sample get viewed with a scanning electron microscope? Well, first of all, it is loaded into a vacuum, which means that it has to be prepped in a specific way. But once that sample is loaded into the vacuum, it is bombarded with a high energy electron beam that focuses on the sample. Once the electron beam hits the sample, it is backscattered off of it, and that backscatter is detected by a detector and amplified, and an image is produced. So there are lots of different samples that can be analyzed using the SEM, but forensics specifically uses it a lot for gunshot residue. If they detect the presence of lead or antimony or barium in a gunshot or in a sample, then they can determine that those are characteristics that are similar to gunshot residue, and that can be used in court. You can also make bullet marking comparisons of the striation marks in a bullet. You can identify different drugs using this microscope. Paint chips can be analyzed from, say, a hit and run victim and a car. Glass fragments from, say, a broken window and a suspect. You can match tool marks, maybe two different types of wood that use the same screwdriver to open up a door jamb. Ink, if perhaps a check is thought to have had a number added onto, like a 0 or a 1 changed to a 9, you can determine if there's a difference in the ink characteristics of one part of the image versus another. And in the case of the study that I chose today, it is used to evaluate the cellular stages of microorganisms, so which could also be applied in forensic entomology, measuring the development of different insects. And all of this information was found from the Thermo Fisher Scientific SEM microscope page, and this image is an image of gunshot residue taken on their microscope. So I mentioned that the sample is inserted into a vacuum. If you notice at the bottom of the image on the left, there's a large chamber where there is lots of negative pressure in there to allow the electron beam to focus on the sample. So that means that the sample has to be prepared in such a way that the vacuum will not destroy it upon analyzing it. So the vacuum will, if there's any of these particles in the sample, they will become destroyed. If there's any super delicate samples, it will simply outgas them. Any liquid that is in your sample will instantly turn into a gas, so that can explode your sample. And because a high energy electron beam is being bombarded at the sample, there's a chance of a charge buildup on that sample. So some sort of conductive material has to be coated over the sample to prevent that charge buildup. The University of Gothenburg has a really good article about how to prep sample analysis, and the Thermo Fisher scientific page describes very specifically how to prepare samples for analysis using their SEM. So they suggest for biologic sample prep, which is really consistent with all sample preps, that the first step is going to be the fixation step, where you need to stabilize either the proteins or the lipids or both. The proteins are stabilized by adding aldehydes to the sample, while the lipids are stabilized using osmium tetroxide. The second step is dehydration, because I mentioned that if liquids are put into the vacuum, they will instantly turn into a gas and thus explode the sample. So each sample must be dried out. And the way that that is done is by gradually adding ethanol or acetate to the sample to slowly allow the water to be pulled from the sample without dehydrating them too fast as to cause shrinkage. Then the ethanol or acetate has to be dried out, and that can be done using just an air dry process or using hexamethyl disalazane or liquid carbon dioxide. The third step of sample prep is to mount the sample onto some sort of conductive material and then cover it in a metal, usually gold, which is conductive to help increase that, to decrease the problems that would occur if a charge buildup occurred on that sample. And then the last step is optional, but if you have a super delicate sample or if you have a sample that is powdered, each individual clump or aggregate of the powder has to be separated, and then the powder needs to be attached to a carbon adhesive, otherwise it would just disappear in the vacuum. And samples can also be coated in a resin to stabilize them and prevent the vacuum from interacting negatively with them. So if the sample is super delicate, that fourth coating and separation step is required. So the validation that I chose is about the parasite Cryptosporidium. It infects the epithelial cells of host mammals, including humans, and it causes the disease cryptosporidosis. Now, as of right now, there's not very many medicines that are able to treat this. And the one medicine that is available is called nitazoxanide, or NTZ, and it requires the body's immune response to get the medicine throughout, to replicate the medicine. So it does not work on children or immunocompromised patients. And the ability to create new medicines to treat cryptosporidosis has been limited because the scientists for a long time had an inability to grow that bacteria on a media that was cell-free or didn't have an immune response. However, there was some research that was showing that maybe it is possible to grow the Cryptosporidium parasite in a cell-free culture. And so the purpose of this study was to prove that it is possible to grow the Cryptosporidium parasite, and their intention is to use SEM images of different stages of the Cryptosporidium life cycle to prove that growth is occurring. So a little bit about the cryptosporidium life cycle is it starts with an oocyte, which release sporocytes, and the sporocytes develop into trophozoites. Then those trophozoites asexually proliferate and turn into type 1 and type 2 merons. The type 2 merons sexually multiply and produce micro and macro agamods, which are going to fertilize each other to produce zygotes. And then the zygotes are going to mature into oocytes and the cycle restarts. So the infectious capability of the cryptosporidium parasite is 10 to 100 oocytes, which is a very small amount, and it is spread via the fecal-oral tract. So a small amount in water can cause contamination. So like I had mentioned, this validation is using the scanning electron microscope to image the cryptosporidium in hopefully different growth stages over the course of 20 different time points. At the end of this study, they found that sporozoites, trophozoites, and type 1 merozoite stages were present in the sample, and they were distinguishable based on their shape and morphology, and their size matched those in previous studies. So the way that they used the SEM to analyze their sample is to first take 10 different time points of growth stages of the cryptosporidium parasite and create a pellet of that life cycle stage using centrifugation steps. So they alternate centrifugation steps with adding phosphate buffer for the first few steps. And then the dehydration process starts with the addition of a 1% osmium tetroxide solution that was allowed to incubate, and then the supernatant was removed. And then the sample is slowly dehydrated with the gradual addition of higher and higher concentration ethanol centrifugation steps. So it started with 30% ethanol, then 50, 70, 90, and 100% ethanol was added to slowly remove the water from the sample. And then the ethanol was allowed to air evaporate off of the sample. And then the sample was mounted in ethanol onto a stub and allowed that ethanol to evaporate. Then once the sample was completely dry, the sample was coated with gold and analyzed on the SEM. So as I mentioned, there were presence of the different life cycle stages, which was the purpose of this study. In order to validate that cell-free culture can be used to grow the cryptosporidium parasite, they proved that with the presence of sporozoites, trophozoites, and type 1 merozoites, that it is possible to grow the cryptosporidium parasite using cell-free media. So this validation, I really enjoyed reading. It had very clean formatting. It was not overly wordy. It was written without the assumption that the reader had a lot of background knowledge. The procedure steps were very clear and concise, and the study seemed easily repeatable with the inclusion of temperatures and centrifugation speeds. And I think they did a really good job of mentioning all of the experimental errors present in their study. However, I think they could have gone into a deeper dive about why other life cycle stages weren't observed, why there were only the 3. And I think it would have made more of an impact had they discussed the impact that the ability to grow the cryptosporidium parasite on or in a cell-free media will have on future maybe medicinal discoveries. These are all of the resources I use, and I hope that you enjoyed. Thank you.